Hour, 10 o'clock. Let's proceed. Welcome to day 2 of the Euglena International Conference. We had a great day yesterday. I'd like to introduce today's session chair, Dr. Barry Seville. Barry's at Trent University, been there for I think now 16 years. I saw Barry. Congratulations.
Wow, I'm old.
Yeah, so I'll hand it over to you, Barry, to begin today's session.
Thank you very much, Scott. I think this is it's going to be a great. One of the things that we've noticed as organizing this conference is the representation that we've had globally. We've got people from the UK, of course, Canada, USA, Japan, Poland, the Czech Republic, Australia, Denmark, Germany, Nigeria, Egypt, Peru, South Korea, Spain, Thailand, and Saudi Arabia, just to mention a few. So today we're going to have a little bit of a slight change in our procedure, because remember yesterday the EIN update, the science update, didn't happen. So that's going to happen today after the first session, and at the end of the day we're going to have the annual general meeting. But realize the annual general meeting is a voting thing for the EIN executive, the science advisory and position paper participants. So the rest of you get to end your day earlier. So that'll be good. Now, I thought it was an amazing day yesterday. I thought I'd quickly summarize what we did. We started off with Allison's talk, which was an excellent overview of synthetic biology. She works with Chlamydomonas, but some of her ideas and knowledge can be shared and perhaps exchanged with the Euglena group. Now, she highlighted key components required for productivity, and those included rapid screening ability, because she noticed that there was a lot of variation in different transformants. So you have to be able to get a lot of them if you can. Define control mechanisms, knowledge of related biochemistry of the organisms. One of the things that she said in answering to her questions was, the more you know, the better. And so that was important, not only for introns and control of gene expression, but also knowing about the biochemistry of the host organism. Then Natalia gave this great talk on extrachromosomal circular DNAs. Now, she identified them in Euglena gracilis, and she indicated some of the molecular and bioinformatic techniques that she used to do so. But she also noted the limitations in her analysis imposed upon us by the not having a well-annotated genome, which is one of the things that we want to move for as a group. And that was followed up by Rodrigo's talk on endo-beta-1,3 glucanase and beta-1,3 phosphorylase, and those enzymes in Euglena and how we might utilize those to learn about them in the organism and beyond, especially how they might be used in polysaccharide degradation. He mentioned in answering to questions about fungal polysaccharide degradation. Session 2 was a great follow-up because Saul gave a talk that was also on Chlamydomonas, and it was a follow-up from Allison's. And one of the things that's interesting, if you know a lot about biotechnology, and I know that a lot of you do, was the recognition by Saul that there's a lot of things that we're going to do. There's going to be regulatory hurdles that we have to go over. And Saul gave a great outline with examples of how we might handle those. So he talked about, first of all, how to transform into the chloroplast of Chlamydomonas so he could do site-specific insertions as opposed to the random insertions that Allison talked about. He talked about different expressions of human growth hormone, antibiotics, and vaccines. And then he talked about all of these things in terms of ways to limit and to control things and to get rid of one of the things that was really cool, getting rid of the antibiotic resistance. All of those have regulatory implications. Great talk there. And then Sahuchai talked about quirky metabolism of Euglena. So this is where we got away from Chlamydomonas back to Euglena. He had this great model of the metabolic pathways in Euglena, and he talked about testing some of the pathways with carbon-13 metabolic flux assays, and he designed experiments with chloroplast bleaching and looking at C2 and C3 metabolism, which was awesome. And then we were followed up by Katsuharu giving us an update once again of their progress on the Euglena gracilis genome annotation, which was a great thing, and it stimulated a lot of questions. So today we are going to have 3 talks in the first session. And the first is by Celine Gauguin. And Celine, I'll let you introduce your talk title. Celine is a fellow Canadian, and she's at the Department of Chemistry in the University de Sherbrooke in Quebec. So with that.
Yeah, okay. Can you see my screen?
Yes, I can, Katarzyna. That's good.
Great.
Take it away.
So, hello everyone. My name is Katarzyna Jankowska, and I'm from University of Warsaw in Poland. And my group is focused on the taxonomy and ecology of autotrophic euglenids. And I wanted to tell you about a project we worked on, which is metabarcoding of euglenids. Before conducting the metabarcoding, the 3 main questions occurred. Firstly, we wanted to recognize the species richness in our samples and compare it to the current knowledge about species of autotrophic euglenids, which is based on available sequences in databases. The second issue was the species composition. How does it look in different samples, and are there any parameters of the environment that have an influence on it, so we could group them. And lastly, we wanted to check whether the changes in composition are affected by the seasons, or is the composition different in different years. Since we wanted to study the influence of different environments and yearly changes, we collected quite a lot of samples. We conducted sampling in 16 water bodies located in 3 regions of Poland, and we did that for 3 years, 4 times a year during the vegetation season of euglenids. We collected samples from small eutrophic ponds, which are the most typical habitat for autotrophic euglenids, and we divided them into 3 categories: fish, field, and rural or urban ponds. After sampling, we harvested the DNA using centrifugation, as it is proven to be the most optimal method to obtain euglenids DNA. Then we amplified the V2 region of 18S rDNA, since it is very well suited to euglenids long and variable sequences. The next generation sequencing was conducted using Illumina MySeq with the reads of the length 300 base pairs. We used the available Silva database and added about 720 curated euglenid sequences, where about half of them was obtained by our group from the environment. All of the bioinformatic data was processed using QIIME2 and R, and we mainly used the vegan and phylosec packages. So, to sum up, we collected 190 samples along with the environmental parameters for every single one of them, which included basic ones like pH and the amount of oxygen, but also the ones like nitrogen, phosphorus, and some more. Using that data, we wanted to find out more about species richness, species composition, and its relations to the environment. But first, I wanted to give you a sense of the obstacles that we had to overcome to obtain reliable results. One of the problems we noticed was the migration of the reads between samples in one run. We sequenced our samples in 4 different runs, and we only noticed that kind of issue in distinct runs. So that makes us think that the reads probably switch tags, which are used to tell apart samples, and it happens on the flow cell during sequencing. You can see it on the example of one of the rare species, Ecology harcoviensis, which was only seen under the microscope in one water body, which is labeled here on the graph as UR19. And you can see that it is also present in many other samples from this run in small quantities, while in the other runs there were no significant amounts of this species. Also, another reason that we think that the presence of this species is due to sequencing mistake is that in this run we had some controls with a known mix of euglenids where we had no Trachelyomonas species added, but the reads also occurred in that samples. To overcome this issue, we decided to delete the 0.5% of reads of every species present in the run from every single sample. After that, the probability that there are random species present in our data due to this issue was much smaller. You can see it on the picture that there are no reads of Trachelyomonas harcoviensis in the other samples than UR19 after deletion. Another problem we stumbled upon was related to the length of the reads. We found out that some of the species have too long reads to be merged while sequencing 300 base pairs from both sides as we did. That way, after trimming starters and taking into consideration needed overlap, we can get sequences up to 450 base pairs. And unfortunately for us, there are some sequences of species that are longer than that. They can have even 550 base pairs, so there's no way for them to be present in our samples. That is why we decided to check whether the situation changes when we only use forward reads instead of merged ones. That approach worked well, mainly for these two shown species, Euglena granulata and Euglena fusca. Lepocynthes fusca. We obtained much more reads of these species without merging. While using only forward reads works well for them, overall it decreases the quality of the analysis. So we have to settle for merging while knowing that this situation occurs. While it worked for mentioned species, there were some other ones that had longer sequences, and using only forward reads did not make any difference in their case. For example, Euglena sanguinea and Trachelyomonas armata. Even though we know that they were present in our samples because their morphology is very characteristic, there were no reads of them. And we think that the reason, besides the length of the sequences, is their high variability, which makes it harder to sequence the fragment we used. Another extraordinary species is Euglena ehrenbergi, which is very common, but there is still no 18S rDNA sequence available, so there is no way to see it in our results. Even though, as you can see, there were some difficulties with obtaining reads, we managed to identify in our samples almost 80% of species with no sequence. That is a great proof that euglenids are in fact cosmopolitan, and they can be found in almost every place in the world. When we look closer at our results, it turns out that the reads of those species sequences comprised about 98% of all of our euglenid reads. Besides that, 1.5% of reads were classified only to genera, but we have checked them, and about half of them are representatives of the group of small facus, which sequences are very similar to one another. So there is a problem with delimiting species based only on sequences. The rest of the unknown reads in our study may occur due to poor quality, of course, or they may represent yet unknown species or even higher taxa. Questions mentioned before made us check whether the number of species is correlated to any ecological variables. We checked how it is related to the type of water body, and as you can see on the box plot, there are significantly more species in ponds located on the fields. The reason might be that it is the most natural habitat from these three types, and it may promote higher biodiversity. Another thing that can be seen here is that the number of species is more variable in the rural or urban ponds than in the fish ponds, probably because the environmental parameters in the fish ponds usually are more stable. So yeah, we studied parameters of these three types of water bodies, and as predicted, pH is closer to 7 in the most natural field ponds and higher in the other types due to substances that may be put in the water, like fish food in fish ponds or sewage in rural or urban ponds. Also, it is not surprising that the range of pH in fish ponds is more narrow because this kind of environment is, as I mentioned, more controlled than the other types. The next parameter is conductivity, and the differences here are not so clear, but the interesting thing is the gap that you can see between values, which is less clear-cut in field ponds, because the changes in parameters here occur more gradually. All of the studied water bodies were eutrophic or hypereutrophic, but the nitrogen entropy, which is based on phosphorus, were higher in fish and rural or urban ponds. So the blooms occurred here more frequently, and which could also be the reason of a smaller number of species. Of course, sometimes in the field ponds, the amount of nitrogen and phosphorus was also rising due to influxes of nutrients from the fields, but in the other two types, usually the amount of them was always high. Okay, so we see that these three types differ in number of species and in parameters, but do they differ in species composition? And as you can see on the picture, yes, but not so clearly. And the reason is firstly, because the data set is too large to obtain good NMDS plots. And another thing is that some of our sampling sites were harder to label as their surrounding couldn't be classified as one type, or for example, it changed in time. So that is why to show the results better, we choose only 2 water bodies per type, which were the best representatives for them. Here you can see that the differences in the number of species between them is quite the same as I have shown earlier. The number of species is the highest in the field ponds, and the smallest variation in the number can be seen in the fish ponds. And as predicted, the NMDS plot looks much better on the smaller part of the data set. And as you can see, the differences in species composition between different types of water body is quite obvious. I prepared another NMDS plot, which is taking into consideration all of the measured parameters of water in a sample instead of the species composition. And the differences are here, but not so clear as on the previous slide. The reason may be that there are some parameters that do not necessarily have an influence on the presence of euglenids. For example, we looked into the presence of manganese and iron in the samples to see if it is related to the number of Cahelomonas and Strombomonas species, which, as some of you may know, possess a lower kaput from that element, but for now, at least, we did not see any relation. And the last thing I wanted to present are the NMDS plots for 2 of the sampling sites, showing the species composition of samples and its relation to the seasons and the year of sampling. And as you can see, there are no visible seasonal changes, but in most of the water bodies we saw, changes in the species composition in years, although it was not seen in the larger scale than just one single water body. So to conclude, we found 153 species out of 197 species with available sequences, and it comprised 98% of our reads. So we see it as a great success of our method of meta-barcoding. Also, we noticed a relation between species richness and species composition to the type of water body, which means that there may be some important parameters or a human influence on euglenids we see in the water. And the last thing is that we do not see any seasonal changes in euglenid composition, but it can change depending on a year. So it means that the things like colder winter or lower water level can make an impact on them. This is all I wanted to say, so thanks for listening, and I am waiting for your questions.
Okay, so thank you, Katarzyna. We're running behind time, so we'll have to move on to Michael and Neil. Are you ready?
Yeah, we're ready, I think.
Okay, take it away.
Okay, I'm just going to put up a short slide deck, but don't worry, everyone, this shouldn't take too long. Can everyone see that screen? Yeah, cool. So this we've been asked just to give update on what the Euglena International Network Scientific Committee has been up to, and this is going to be a double act from the co-chairs, which is myself and Michael Ginger. So just to say that the scientific committee doesn't do any science, so our role is really to try and just facilitate some activities and hopefully make everybody else's life a little bit easier in the process. So very briefly, who we are. This is the scientific committee. I hope, relatively representative of the distribution of the Euglena community internationally, though as Barry pointed out at the beginning of this session, it's a very international meeting we have here, but we have representatives from the UK, the US, Canada, elsewhere in Europe, Germany, Poland, Czech Republic, and the representatives in Japan. And we meet every couple of months. And these are the main things that we've been going through over the last year is the position paper that has recently come out, and Michael's going to say a few words about that. We've been coordinating some of the genome efforts. I'll talk about that, and then we've been trying to put together a species list to inform genome selection for some of the global efforts to sequence eukaryotic biodiversity. But first of all, I think, Michael, do you want to say something about the Euglena position paper?
Yeah, okay, thanks, Neil. And I'll talk very quickly because I'm aware that people maybe want to go and get a cup of tea. So just to update briefly, thank God's pushed forward, and he's led the authorship of the EIN position paper. It's coming out later this month in Biology Open, and it's going to be accompanied by a wider press release. For any that are unaware, then this concise position paper states the network's objectives, which principally are to gain a greater understanding of euglenoid biology through genome sequencing of many diverse euglenids, and from there to look for opportunities to translate any of the biology that's discovered with industry. The position paper also states the EIN's commitment to open science wherever possible. I'm going to pass back to Neil in a few seconds' time to update on genome sequencing. But I guess an obvious question is what we also might do next to continue to raise awareness of the EIN. Perhaps somewhat flippantly, the obvious answer might be to keep reiterating the mission to as many as possible of the EIN. But one thing we're looking to do following this meeting is to provide a special issue around euglenoid biology for publication during 2023. We have in principle a yes to a special issue from one relevant journal. In case there's just any 11th hour change of heart from that journal and a retreat from a yes in principle, I'm not going to mention the name of the journal, but it's something that we hope to update on very soon, and we will be writing to all that are attending this meeting to see if there is interest to join said special issue, which I hope to update on further very, very soon. I'm going to pass back to you now, Neil.
All right, thanks very much. And yeah, this is really, I would just, I should thank God again. I think we just do that on a daily basis for really pushing this forward. I think this paper is going to be really useful for being able to show to funding agencies and others about how well coordinated the community is. So in terms of genome efforts, so genomes, as you know, are like buses. There's been none for ages, and then several come along at once. And we're aware of around 4 different efforts around the world. And what we want to do is try and bring those different labs together to ensure that what we get out of this, all these efforts, which is great work for all the labs that have been doing this, a single agreed community reference genome of Euglena gracilis, which obviously is the model which most people will be working on. So what we've had a meeting of the various groups involved recently, and what we want to do next, and what Ross Lowe, who's the scientific committee secretary, has been doing is going to put together a data sharing agreement to make sure that all of the groups involved can share their data. Hopefully, you'll be able to use each other's data to improve their assemblies, and potentially their RNA-seq data to improve the annotations. And what we'd like to be able to have is an agreed sort of community single reference genome for Euglena gracilis, so that we could be working off the same reference across all of the labs. But of course, there are many other efforts now around the globe. So due to sort of the numerous Earth Biogenome Project consortia that are taking off, in the UK, there's the Darwin Tree of Life project. These sequencing consortia are actually looking to get samples for their pipelines for their genome projects. So I know in the UK, they're looking at using culture collections of any species which have a UK origin for full genome sequencing. In anticipation of this, and this probably taking off elsewhere in the world, we have been putting together a list of all known strains and species which are in culture collections, so that we can readily provide these or information about these to the genomic sequencing consortia. So some of these strains will have been identified by barcode sequencing. Some could be grown axenically, some haven't been grown axenically, some there'll be good provenance information, some there won't be. We've tried to record all of that as best we possibly can. So identified 97 different species which are listed in culture collections, about 63 of these 97 species which we've which are listed, and 63 of these are in collections. But like I say, so hopefully that will be able to inform future genome projects and accelerate the number of genomes that are coming out in the public domain going forward. But obviously, we welcome more information from you. Currently, there. Sorry, I've got the wrong slide. Gone backwards, not forwards. we would like people to let us know about any genome sequencing efforts they're aware of that we may not know about, so that we can make sure that people don't end up repeating each other's work. If there are strains or species you hold that are not in public repositories, that's also useful to know, especially if you've got phylogenetic information from barcodes that can help place those. And do let us know anything else you think that the EIN scientific committee can help facilitate or coordinate between labs. Emails are there. We look forward to hearing from you. Thank you. That was brief enough and everyone's got time for a coffee.
Thank you so much, both. That will end our session. So please go have coffee or tea and we'll rejoin in, let's say, 5-10 minutes. Is that okay?
Cheers. Thank
Wonderful.
you.
Thanks, everyone. And thank you, Barry.
You're welcome, Scott.
Yeah, excellently chaired, Barry.
Okay. Welcome back, everyone. I just first would like to introduce Emma Kazeki. Emma will be moderating the second session. Emma is a PhD student in the Emory and Seville lab at Trent University. She is studying metal uptake in Euglena. And she was also first author on last year's conference proceedings in the journal Protus. So I'll hand it over to Emma now to moderate the second session. Thank you very much. Over to you, Emma.
Thanks, Scott. And thank you, Michael, for making me first author on that paper, because it's definitely good for my academic career. And I'll just start by saying that I feel entirely validated based on all of our speaker talks this morning. So thank you for making me feel like my PhD journey is on the right track, because everything that we discussed is what I'm also trying to do, except in Euglena mutabilis. So knock on wood, hopefully I'll have all of that published by 2026 and get out of here as a doctor. So that's really exciting. Thank you for that. And now it is my pleasure to introduce Dr. Michael Liebert from the Friedrich Alexander University in Germany for his talk. And oh, there he is. Hi, Michael.
Hi.
You're all good to go.
Yes. Okay, thank you so much and for the nice introduction, and greetings from Houston. I am attending another conference right now and I'm sitting in my hotel room. Technological wonders make it possible. So what I'm trying to give you is a little bit of an overview of what we did for the last 20 years in the model system for the analysis of environmentally controlled signal transduction chains and closed life support systems. It's about 10% of the stuff we are doing. And I will try something totally different. I will give the first more or less classical view, the classical facts we assembled over the last 20 years. And then I will give you an insight of what we are trying to figure out right now. And then I will talk a little bit about life support systems. I hope this is going to work. And I hope I'm finished in 22 minutes. And I have my timer set. So let's start. So what we have over here is the professional gravity sensing cells. This is Euglena gracilis, which is moving forward with about 1 to 2 cell lengths per second, means about 50 to 100 micrometers per second, and it's rotating around its long axis with 1 to 2 hertz, which becomes interesting and is probably necessary for the detection of light as well as gravity. So in low light conditions, so when the sun early rises, it shows a positive phototaxis, which turns around, and when we have highlight conditions, and shows a negative phototaxis. What we have when they are not aligned with the gravity vector is that they show a negative gravitaxis under some circumstances, but this is very seldom. They also show so-called positive gravitaxis. Now, when we started out, or what was an enigma for 150 years is the question of whether it's a passive or an active orientation. So it's a boy, or is it something which is really actively oriented, so it's sensing and actively moving in the direction of the vector. And my coworker, which I put on the title slide, this is Peter Richter, came up with a really crazy experiment. And what we did is in a Texas experiment, which is a sounding rocket experiment where we have 4 to 5 minutes of weightlessness. We put in actively moving cells and some which we fixed with liquid nitrogen, so they preserved the cell form, but could not. They were dead cells, so they couldn't swim anymore. So and then we applied different amounts of acceleration. So it's 0.1 and very low over here, 0.2, and then there is a 1 times. So you don't have that much time. So the 1 time is the ground control. Now 0.1 might become interesting because it's a threshold for the gravitactic sensing. So when we compare the unmotile with the motile ones, and we're looking here for the slope of the oriented cells, so how fast they are reorienting their long axis in parallel to the acceleration vector. Then we see that about 20% of the reorientation comes from a Boy effect, which I was able to determine beforehand in a paper which I published, I think, in '96, something like this, where you could determine that about 20% of a Boy effect was to be considered to add to the active one, but 80% of the orientation is in fact an active orientation in respect to the gravity vector. So this solved that enigma. Now the question was, how could they do that? I mean, these are small cells after all, and what we have is that there are no obvious sedimenting cell organelles. You might remember from Biology 101 that in the roots of plants, they are sedimentic particles, so stabolites, essentially amyloplasts, which are sedimentic according to the vector, and they are triggering the response. That becomes increasingly difficult if you have forward moving and rotating at the same time. So this didn't sound right, and so what we did is, first of all, we determined the specific density, which is about 1.05, so it's about 5% higher than the surrounding medium. And this again gives us a force which can be applied by the sedimentic cell body of about 1 piconewton. But yes, what we also saw was that below a threshold of 1.02 grams per milliliter, we didn't see any gravitaxis. Now, when we did these experiments, we did also experiments where we changed the specific density of the surrounding medium, which is normally 1.00 grams per milliliter. This is essentially water. And what we see over here is a circular histogram, which shows the bar length, the number of cells going in a certain direction. So what we have over here is a very nicely oriented population of cells. And this one over here represents about the measured of 1,000 to 1,500 cells. Now, if we increase now the specific density of the surrounding cell of the surrounding medium, nothing much happens. It's a little less precisely oriented, but at 1.02 we see nothing. But if you approach the specific density of the cells, we have an 1.04 and 1.06. We do not see any orientation anymore, and it changed the sign if we are well beyond the typical specific density of the surrounding medium. So it seems like that the density difference between the medium and the cell body plays a crucial role. Now, what else? Now, of course, you look for calcium, and what we did over here is a calcium crimson intoxication of the cells, so to speak. And what we see over here is relative fluorescence. And what happens over here is that we, again, another crazy experiment of my coworker, Peter Richter. We put it on a record turntable, and let's them orient and then turn the table by 45 degree or by 90 degree or by a 180 degree, and look for the reorientation. What we see over here is that we have an increase of the calcium fluorescence. And this comes very nicely together with the measurement of the gravitactic orientation, which is given here in the red boxes, in the red squares. The R value is a statistical value telling you how good the population is oriented. Now, if you're talking calmodulin, if you're talking calcium, you probably think about calmodulin. And turned out later on in working together with my English partners, and especially, thank God, we found out that there are plenty of more of calmodulins. And the first one we looked at was known as very long at 1992, Toda et al. Published that there was a calmodulin on the protein level. Then we sequenced that gene and found 2 more calmodulins in sequence analogy. And then we did inhibitory RNA, and I think I don't have to go into too much details about the technology. Anyway, it's this is depleting the existing mRNA of that protein, and so you have less protein. That's the hope. You have to verify that, of course. Unfortunately, Cal1 swimming motility was lost, and metabolic motility persisted, and cell division was affected. So what that means is, after recovery from RNA, gravitaxis was again visible. If the cells are not moving, you do not have the response you can look for. So that's not of great help. But what was of great help was that we have here the control cells. This is the control culture. There are very limited number of cells after the inhibitory RNA method. And when you look for details over here, and I think, yes, here you can see it, we have a problem here with cell division. You know that these guys are removing their flagella, and then along the long axis, they are going to divide. So something is wrong, and this calmodulin has something to do. And sometimes you have to simply be lucky. And the next the Cal2, which we analyzed with RNAi, you see that the RNAi that's here in the circular histogram, there's no orientation anymore. The control is just fine. After 30 days, the control is still fine, and the RNAi, the treated cells against Cal2 are still not showing any gravitaxin. It takes about 6 weeks that the response is coming back. So we are diluting out the inhibitory RNA. Now that was nice. And what else did we do? Then we looked for cyclic AMP, another second messenger. And again, Peter did experiments over here, and these are with syringes, where you have cells, where you can cell stimulate in different ways, and then fix them at a very fast path. And what we get over here is that we have a 100 milliseconds range for the determination of the amount of the cyclic AMP. And what we have as stimulants over here is not gravity. It's, in this case, it's light. And let me remind you that they have a very interesting photoreceptor found in Euglena, which is PAC alpha and PAC beta, which are photoactivated adenylate cyclases. So they produce in the photoreceptor cyclic AMP. And what is nicely shown over here is that after 100 milliseconds, you have already a starting building up concentration of cyclic AMP. In contrast to many other photosensitive organisms, the response of Euglena is relatively slow. So it takes a couple of seconds before they're really starting to react. At least at the stimulation level we used over here. What is shown over here is how the PDA activity is done, and we looked for the concentration over here. This is approximately the same status over here, and looked what is happening in the darkness. So we have continuous illumination over here, and what happens in the darkness, and you see at least a slight the slight idea of that the cyclic AMP is going down, and this should be the phosphodiesterase activity. Activity should be spelled with C, but I'm sorry for that. The same actually can be shown in again, Texas experiments where we look for the cyclic AMP, and we have here the level after about 1 to 2 minutes in weightlessness, and go here for just below the threshold and just above the threshold over here. And we see a dramatic increase in the cyclic AMP. So cyclic AMP plays a distinct role in phototaxis and a distinct role in gravitaxis. Let's see what else do we do. When you look for the cyclic AMP, so we knew by inhibitory experiments that phosphorylation does play a role in phototaxis and in gravitaxis. And then we were lucky again, so we tested a couple of protein kinases, kinase A, which are cyclic AMP dependent, and that was inhibitory RNA. And what we saw over here is that these are the controls, these are the cells which were inhibited, and that at least in the beginning, you do not have any orientation anymore. Again, circular histograms giving the amount of cells moving in a certain direction. But after 22 days, it comes certainly back. Now, that was for gravitaxis, but to our fully surprise, it was also true for phototaxis. So controls, and here the inhibitory RNA, and we're looking for negative phototaxis, which is much easier to use in these kind of experimentation, and no response over here. So it looks like that we got the missing link between phototaxis and gravitaxis. All right, so where are we now? That was the model when we started. So this is about 5 to 6 years ago, and we have a cell not aligned with the gravity vector, and we have a calcium influx over mechanosensitive channels, which I did not mention up to now. We have the calmodulins, which bind the calcium. The calmodulins, in fact, then activate an adenylate cyclase, which we could not identify up to now. And we produce them by the adenylate cyclase, cyclic AMP. And this is somehow interacting with the flagellum, the trailing flagellum over here. You know, remember that there are two flagella. One is not coming out of the position over here, the reservoir, but the other one comes out, and it is essentially the one responsible for the movement of the cells. At the contact point over here, we have this paraoxynormal body which contains the photoreceptor. Now, then we change the beating pattern of the flagellum, and that got a reorientational movement. Some more stuff which we need, the esterase, so the PDE, and we have a protein kinase, the protein kinase A I already mentioned. Now, that was 5 years ago. Now, what did we do in the meantime? We developed some antibodies, which is not easy with Euglena because I mean this is probably oils to actins, but because they are so close to trypanosomes, many of the rabbits which you could lose for production of antibodies already have trypanosomes infection for some reasons, and so they have a lot of antibodies against many of the proteins of Euglena 2. So this is the fluorescence image over here. This is the overlay over here, and this is the cropped and enlarged image. And this is the apical portion. So it is very close here, probably located in the reservoir, and which nicely fits with our expectations. What was kind of a little bit of surprising is that we have a knockdown. When we have a knockdown of PAC alpha, then pKa is downregulated. And so there's a direct link between polycistronic expression of the PAC alpha and the pKa. So no PAC alpha, pKa is downregulated. That means also that just a second, that gravitaxis is then impaired. Phototaxis and gravitaxis is impaired in the moment when we have PAC alpha knockdowns. And what we here, the depletion of pKa after downregulation of PAC alpha, this is the controls which we have over here with these cells with antibodies against PAC alpha. So that ensured us and make us more confident about that we have that we have something with PAC alpha and the pKa, and that we in fact are coming to the point where the interaction between gravitaxis and phototaxis becomes eminent. Now, what we did further on is what is shown over here is use these two hybrid assay, and we found then a protein which is Euglena gracilis, and it is a DUF protein, which is domain of unknown function, which is kind of not really helpful. But anyway, when we used antibodies against these DUF protein, this DUF domain, then we saw that it's located here in the flagellum, which makes it complicated in the interpretation of what is really happening. So the knockdowns of these proteins resulted in something which, depending on the C-terminal targeting or N-terminal targeting RNAi, that we have a loss of gravitactical behavior. But for flagella, it's a complicated mechanism in there. So if it might well be some follow-up effect and not a direct effect. But my co-workers explained to me that it is really a direct effect, so I believe them. So it's seems to be directly involved. Now we only have to find what it is. Now, the second thing is what is shown over here is the CAM2, which I showed before, is important for the gravitactical behavior. And it's here still in the fluorescence, the greenish fluorescence. It's obviously also here in the flagellum. But it's also randomly distributed. I don't see any direct pattern in there over the cell body. And that's one of the things we are puzzled about, and we have no idea what it really means. Now, in order to further evaluate this stuff, we were always hampered that we do not have a transformation system for Euglena. And a couple of years or 3 years ago, I guess, and it was published last year, we found a system with Agrobacterium plumifasciens, which allowed us to do the nuclear transformations. These are the vectors we are using in there. And when you look over here, hygromycin is a very effectively but slow acting slow-acting antibiotic against Euglena, and this is the control. No hygromycin and the control cells, and these are the cells, the control cells with hygromycin after, I believe it's 2 or 3 weeks. I'm not quite sure. And these are the cells which are in fact actively transformed by the method over here. You can also do that with gas, of course, and gives nice little stainings over here. And it works like a charm. It's a very effective method we have in there. And of course, we did the successful transgene insertion analysis, which is shown over here. Now, CRISPR-Cas is everywhere, and also we did it. And what we did is control. And again, my timer now tells me I'm out of luck. And just
you do have a few extra minutes, Michael, because we started early. If you wanted to keep going.
yes, I will. I try to figure out how to stop my timer. All right. This is light direction, and these are CRISPR-Cas knockout mutants, clearly showing that there is no phototaxis anymore. And when you look for the content of cyclic AMP, and then I showed that before, when we eliminate it, we see an increase of the cyclic AMP, and this is gone over here in this kind of experiment. And this is paralleling what we did for RNAi knockdowns, but in contrast to what we see, what we have in the inhibitory RNA, this is really, there is no protein anymore, there is no discussion, and there's no doubt that it's really an effect of this method. At this point, so that's where we are right now. So we are still working in AS. Unfortunately, we had a gap of about 3 years where where the grant agency was not supporting our molecular biology work anymore. It's coming up now again, because the new grant is now giving us a leeway to do what we always planned in further analysis of the gravitaxis and the phototaxis, especially in the interaction, because that is something which is very interesting, not only for Euglena, but also for many other organisms. How our signal transduction chains are interacting. Now, what we this is just one example of what we did in the meantime. So we are not bored. We are away from the streets, and and this is the Eucopus project. This Euglena bacillus combined regenerative organic food production in space. A space experiment which is testing biological life support systems under lunar and Martian gravity. And this is essentially a satellite which is rotating around its long axis and giving you 0.16, so it's moon, and or 0.38 times g, the acceleration, which we then can use for testing the whole stuff. So what we have over here is a control and data management system, and I need to come over here, and it turns out that the system becomes increasingly difficult if you have many components. What we have here is a greenhouse, we have a crop filter, and we have algae containers. Algae is, of course, first of all, this is I have many discussions with my colleague about whether Euglena is really an algae or not. We settled for protest. I think that's fine enough. At least it's green. We have this greenhouse where we are growing tomatoes in there. We have a crop filter with a bacterial filter, which is transforming urine so essentially urea to nitrate as a fertilizer here for the greenhouse. And it leads in a closed environmental systems, you need a lot of control. So what we have is 30 temperature sensors, 3 flux sensors, we have fan sensors, we have filling sensors, we have pH sensors, O2 sensors, we have an integrated ion chromatography, we have an integrated PCR system, we have a Ramis control, Ramis radiation measurement device, and we have, of course, an illumination, we have palm measurements, which is a contactless possibility to measure the photosynthetic performance of the cells. And the crop filters also have these O2 measurement and pH measurement. We need obviously a filling system and so on and so on. And that's the general scheme. Over here is a water tank. We have here the crop filter, and we have here the greenhouse. Excuse me. Here is the crop filter, and here are the algae container, the crop filter, the greenhouse, and we have fans in there. And I would be more than happy to explain why we need fans in the greenhouse in weightlessness or less gravity over there. That's me. It's a colleague of mine in DLR Cologne, and that is one of my colleagues from Erlangen. This is Sebastian Strauch, who is doing some work on this over here, because I think what is exceptional is that we build our own hardware and do our own biology stuff. So we know we have everything under control, which is very nice. And what you also need, and this is my last slide, I promise almost is my last slide. What you need in these kind of experiments is that you often have to close up your system a couple of months before you really can fly. This is no problem for seeds, but what about the algae? And what we found in the literature is that Euglena is forming nicely encysted forms, palmellar forms, and which can sit in the on wet cotton wool for 9 months. I think Peter might correct me, but it's now for more than 2 or 3 years that they are sitting on cotton wool. And what you see is here the culture grows. Coming back after 9 months, but it's the same for 2 years and for 3 years. So you can store them almost indefinitely. The recovery of photosynthesis, gene expression after the recovery, and recovery of gravitaxis, and it comes back nicely. So you can store Euglena for almost indefinite times, as far as we can tell right now. These are the acknowledgements. It's finally really my last slide. And don't forget for the funding agency, this Deutsche Agentur für Luft- und Raumfahrt, DLR. Thank you so much for your attention, and I'm happy to answer any questions if I have any more time. Emma?
Amazing. Thank you, Michael. We're going to save questions for the end. So if you could please stop sharing your screen, we'll transition to a fellow Canadian colleague, Dr. Von Mangel at the Brock University in Canada. So Von, whenever you're ready.
Thank you so much. Special thanks to Scott for giving me a friendly reminder that I got the timing wrong for this. So thanks, Scott. I actually had a little surgery yesterday, so I'm a little bit woozy still. So I apologize in advance. Everyone can see my screen now. Okay, wonderful. Well, thank you. I'll try to stick to our time here, but today I'll be talking about Euglena gracilis and specifically some of the work that we've done for the metabolomic side of Euglena gracilis with respect to mercury biogeochemistry. So once again, my name is Dr. Von Mangel. I'm an assistant professor at Brock University here in Canada. So just to kind of give you a kind of brief case study, in 1932 in Minamoto, Japan, there was severe mercury spilling that was happening. And at the time, we didn't really understand the biogeochemical implications and the human health consequences of mercury in the environment. What ended up happening was that around 40 years later, once we identified methylmercury being the main source of many people having neurodegenerative disorders that were eating the shellfish and fish in local aquatic systems, that the company here was ordered to pay back close to $75 million to all of the victims of this disastrous event in human history. So to this date, mercury is a common problem that we're trying to help alleviate in contaminated settings, and mainly mercury.
Vaughn, it sounds like we lost your audio there for a second.
Yeah. No.
Perhaps try reconnecting, Vaughn.
He does look very frozen.
Is everybody still there?
Yes.
Yes, of course.
Okay.
Yes.
Okay, Vaughn, try reconnecting perhaps.
Everybody?
No, just
No,
Vaughn.
just Vaughn.
Vaughn, just Vaughn. Sorry. Yes, please don't go.
Sorry about that. Can you guys hear me now?
Yes, we can hear you again.
I'm so sorry. I am going to continue.
And he's back.
And you guys can see my screen?
Yes.
Yes.
Thank you.
So sorry about that. Okay, hopefully that doesn't happen again. So kind of continue.
It happened again.
Fuck. Okay.
Still no sound, Vaughn.
Hello, you guys still can't hear me?
We can hear you now.
Okay. And you guys can see that?
Yep.
Okay. So one of the main problems with mercury is that in its inorganic form, it's still toxic, but really it's when it's methylated by methylating microorganisms. So one of the rate limiting steps for mercury is to determine whether mercury gets converted to methylmercury is how microorganisms can uptake this mercury. This is kind of where we thought about including Euglenoids in this project, because studies have shown that when organic matter originating from algae and other microorganisms is present, there are actually cases where more mercury gets converted to methylmercury. So we thought we'd kind of explore this a little bit more, and because Euglenoids has unique properties, they've been shown to produce high proportions of proteins, unsaturated fatty acids, and waxy esters. We began to hypothesize that maybe the chemical composition of Euglena might elicit different effects from a mercury biogeochemistry perspective. So this kind of drove one of the papers that I'll be talking about today, where we looked at how varying the types of organic material from different algae influences whether or not microorganisms can uptake mercury. Other studies have talked about organic matter, especially from algae, stimulating this production of methylmercury, but until now, we didn't really understand why. So to use this and to test this, really, we kind of adopted a biological reporter system for our environmental applications. Essentially, what happened here is we developed this plasmid and inserted it into an E. coli cell, where when we vary the environment of the metal, so in our case, mercury, with different organic substrates originating from different algae, we get a proportional fluorescence response that is directly indicative of how much metal has made it inside of the E. coli cell. So the nice thing about this too is that this can be done in both aerobic and anaerobic settings. And in particular, for particular importance are these kind of anaerobic settings where methylation is more prone to happen. So what we did here is we cultivated 5 different types of algae and protists. We looked at Synodesmus, Chlamydomonas, Chlorella, and then 2 different types of Euglena species, Mutabilis and the Gracilis species. So what we did is we increased how much organic material was exposed in around this algae in both aerobic settings and anaerobic settings. And we essentially found that as we increase more carbon to the system, there is a rather exponential decay or decrease of how much mercury was making it into a bacterial cell. This was the case for a lot of the phototrophs like Synodesmus, Chlamydomonas, and Chlorella in both aerobic and anaerobic settings. Now, things began to get a little bit different when we looked at the Euglena species. We saw that with Euglena mutabilis, it elicited a similar response where there was that exponential decrease as soon as low as 0.1 ppm or milligrams per liter of carbon was present. But this same inhibitory effect was not being pronounced with Euglena gracilis. You'll notice that here on the x-axis, we went all the way up to 30 milligrams per liter to also see if this was just a concentration issue, but we began to see that the same level of inhibition was not present with Euglena gracilis in both aerobic and anaerobic settings. So this led to the question as to what is happening with Euglena gracilis. Why are the molecules that are being released by Euglena gracilis, they don't have the same inhibitory effect from a mercury perspective? So this is where we decided to probe some of the additional compositional properties of organic matter, specifically from Euglena gracilis. So to accomplish this, we used high resolution mass spectrometry to obtain molecular level information at a unprecedented level of resolution that is ideal for non-targeted metabolomics to a high degree of accuracy. And we also use field flow fractionation and size-based separation to determine other metrics of the carbon other than just concentration to see how things like size of all of the molecules actually impacts the ability for it to bind to this mercury. So concentration really isn't the entire picture here. Some of the molecular properties of the carbon were determined using high resolution mass spectrometry, where we get very discrete peaks that provide a high level of detail for compositional assessment. And we can use this kind of untargeted snapshot of everything that's being released by Euglena and other protists to really look at how the chemical profile and metabolic profile changes between these different algae. So the first thing we did here is we looked at molecular weight to tease that apart. And essentially what we found was that size matters. The larger molecules that were being released predominantly by the phototrophs tended to inhibit a lot of the mercury uptake within these cells. But for Euglena gracilis, they didn't have the same level of size characteristics of the organic material that was being released. So Euglena gracilis was essentially releasing lower molecular weight and smaller material. We broke this down a little bit further with the high resolution mass spectrometry to show key patterns that were unique to Euglena gracilis. So here, yellow is Euglena mutabilis and purple is Euglena gracilis with 5 or 6 key metabolites that were significantly and statistically related to how much mercury was being uptaken aerobically and anaerobically. And we found that Euglena gracilis released significantly higher proportions of amino acids like glutamine and serine that also related to increased mercury bioavailability within these different E. coli cells. In anaerobic settings, things like cytosine, glutamine, and polyamine had linear relationships to increase mercury uptake, once again, largely found in Euglena gracilis, whereas the low abundance of carboxylic functional groups, so COOH, also related to a reduction in mercury uptake. So not only are Euglena gracilis actually releasing more of these specialized amino acids that bacteria seem to like, but that also the amount of COOH potential binding sites for mercury to be found on are rather low. So that was one particular study, and in a more recent study that we worked on with Dr. Slink again and Dr. Barry Seville here, we looked at how if we grow Euglena with mercury, really, how does this affect the metabolome, but also the transcriptome level? I realize I'm a little bit late, so I'm going to try to go through these rather fast.
You're okay, Vaughn. We're going to give you until 12:20, okay?
Okay, thank you so much. So to begin, we looked at how the metabolic profile changed before and after growing these Euglena cells in the presence of 5 micromolar concentrations of mercury. Essentially, what we found was that these control cells exhibited very different metabolic profiles than when these Euglena cells were grown in the presence of mercury. From an extracellular perspective, we found that amino acids like and polypeptides like glutathione, trisaccharides, glycines, and serines were significantly upregulated, whereas within the cell themselves, other sulfur-bearing metabolites like cysteine, glutathione were also upregulated after Euglena was growing in mercury. We then also did transcriptomic analyses of Euglena gracilis. So we isolated the RNA, we did de novo transcriptome assembly and differential gene expression to see if these differences at the metabolome level were also being conserved with what we're seeing at the transcriptome level. Essentially, what we did here is we found that there were a few key transcriptomic pathways that were upregulated, and to link that to some of the metabolome level shifts, we did integrated transcriptome analyses, where essentially we found conserved molecules in the metabolite pool, as well as the transcripts that were significantly enriched after mercury exposure. We found things like overall metabolism of amino acids, translation, cellular responses to external stimuli, DNA repair, and detoxification of reactive oxygen species. So really what this means is that when these Euglena are being exposed, and Euglena gracilis specifically is being exposed to mercury, it amps up its amino acid production. And interestingly, there was a detoxification of reactive oxygen species. When we put all that together, what we hypothesize is actually happening here is that Euglena gracilis has a very unique way of dealing with this external mercury stress. Really, this mercury that comes into the cell can induce reactive oxygen species generation at the chloroplast, at photosynthetic light harvesting systems, and really to help mitigate this reactive oxygen species stress that mercury is inducing, the Euglena cell amps up its amino acid and peptide production, especially cysteine and glutathione, key amino acids and peptides that have sulfur-bearing binding sites that can bind to mercury and make it less toxic. As a result, what we think this feedback between mercury exposure, reactive oxygen species generation, and amino acid and peptide production, this leads to the binding to mercury to make it a bit less toxic, and then it's re-released back into the environment. So up until now, there really hasn't been much work that looks into how the metabolite pool of Euglena specifically responds to some of these external stressors. So we've pitched an idea here that really it's Euglena's unique properties of being both photosynthetic but also heterotrophic that allows it to have this unique reactive oxygen species kind of mitigation and very sensitive to reactive oxygen species stress. And really, I'd like to finalize and finish the presentation by talking about some of the different patents that we have as a result of some of this work. So myself, I was a student of Dr. Celine again. So we've patented some of these technologies specifically with looking at how we can use different fractions of organic material and apply that for binding metals, as well as how we can encapsulate some of these biomasses of Euglena, both dry and more of the solvent kind of organic molecules for metal binding as well. I'd also like to acknowledge and thank all of my collaborators, University of Toronto, University of Ottawa, and University of Sherbrooke and Trent. And I should also mention that in the next few years, we're planning on using Euglena gracilis in more of natural systems. We're looking to see the effects of rare earth elements in landfills and how we can help use Euglena gracilis to alleviate some of the increased metal loadings that we're seeing in runoffs from landfills. So I apologize for the technical difficulties, but thank you.
Awesome. Thank you, Vaughn. Okay, so let's get into some questions. I see we've got some in the chat here. If anybody has any questions, throw them in the chat or feel free to raise your hand. I've got a question for Michael. Is your agrobacterial mediated transformation able to apply for metabolic engineering, specifically for the expression level
I guess
your
so.
CAMV promoters?
Yes. Caulobacter. What is it? Caulomosaic virus. CMV. Okay. Yeah, I guess. You always have to try. There's no definite answer. Every transformation is a adventure.
And that is the science rabbit hole. I've got another question for you here, Michael. Have you considered overexpressing the same genes in Euglena? And what might you expect to happen if you did that?
Yes, we did. Which genes we are talking about? When we talk about PAC alpha and PAC beta, Foster et al. In 1991 published a theoretical paper about that the photoreceptor is 10 to the power of 7 molecules in there, and it's already overexpressed anyway. We didn't thought about it, but it might be a good idea. For example, the chimodulines and the pKa, which would be very nice to see what's happening if we are overexpressing. But as I mentioned, for the last couple of years, we were placed on the sideline because we didn't have any money to do more experiments with that.
That leads into the next question that was asked, which is, again, for you, Michael, for the Eucropus project. Now that you have some funding, do you have plans for another space launch? And have you considered using
we are
talking
serious money. We are doing a lot of space launches, but we are talking serious money for projects like Eucropus. We are talking about 10 to 15 million euros or dollars. Doesn't make any difference right now. This is hard to get by. What we are planning to do is some smaller projects, which will not involve tomatoes, probably, maybe yes, but probably not, but may include wine plants, which is another project we are doing a lot of research on in the last couple of years.
Amazing. Vaughn, it looks like we've got a couple questions in the chat about your experiments and model testing, specifically with the deployment in natural systems, if you wanted to speak to that.
Yeah, of course. So in some of these natural systems, we're looking at working around the landfills of the Ottawa region. So a lot of these landfills specifically have rare earth elements kind of leaching out of electronic waste. So the idea is to kind of collect samples and to test and see if Euglenoids can also have the similar uptake response, how Euglenoids responds to some of these metals. And really, if we can use some of the technologies that we've built on with manipulating the organic material that is being released by Euglenoids to help facilitate the removal of these metals from the landfills.
Scott, it looks like you've got a question. Do you want to jump in?
Yes, Michael, this question's for you with regards to Eucropis. I was wondering, have you ever considered to take the plants out of the system and just use Euglena as the food source? So you could grow the Euglena heterotrophically on perhaps...
Somehow astronauts are kind of hesitant to eat a lot of Euglena algae or even Euglena. It is never was really considered also by NASA doing that. There was in the 90s and beginning of 2000, there was some ideas about using algae over there. The problem with the algae is that that the relation between the water and the biomass you have is not good. So what you're probably looking at is having some two-dimensional equivalent of a leaf and grow the bacteria, the algae on that. And then you can use much less water. So the weight constraint is then limited. That's probably the way to go. And I always consider that the nutritional value of Euglena is very high, but it's also the possibility if we now have a transformation system, or even with CRISPR-Cas a knock-in possibility, that we can then look for stuff which is really needed on long-term exploration mission. So they can produce essentially everything. Is that sufficient?
Yes, that was great. That was great. Thank
Okay.
you. Well, I guess a follow-up to that question would be, have you tried using any other species as opposed to Euglena gracilis?
No.
No? Okay.
This is our pet. So why start another thing? I have a colleague of mine, Gil Kramer, who's working next door. He's using Chlamydomonas. But I always think that Euglena is more cute than Chlamydomonas.
Yes, we all think the same thing.
See? All right.
I'll throw a question out there for anybody to answer if they feel like chiming in. This is plugging myself. I work with a Euglena mixotroph culture it has Euglena cells, it has a bacteria, and it also has a fungi hanging out with it, and they've evolved to grow together. So I guess when we're on the topic of using it for real world purposes, has there been any further investigation from any of the groups here that are looking at creating these systems, synthetic systems for things like heavy metal removal and bioremediation? We know that it's getting pretty popular in the literature. So if anybody else is doing it, that would be kind of cool to hear about.
We have one experiment done a couple of years ago when we have big fermenters, 20 liters or something like that, and we tried to grow them exenic, and it never worked for a long period of times. I mean, I'm probably not the only one who has the same experience. Work more dirty, and you are happy as can be. We had to stop at one point or another after 3 years or something like that. The Euglena was happy, the bacteria were happy, other little protists were happy, and this system is much more productive.
Thank you, Michael. I appreciate those comments, because I also have tried to spend the last 2 years separating it, and it is just not working. So
no,
I agree, it is very productive. It looks like we've got another question in the chat. Within Euglenoids, how exceptional is Euglena gracilis in terms of heavy metals, and has this been surveyed across many species or just a few? So Vaughn, that might be speaking to the research that you did.
Yeah, definitely. So Euglena gracilis, actually Euglena mutabilis has been one of the best organisms that is most tolerant to different metals. So the only real exception that I've scoured in the literature is copper. So for whatever reason, Euglenoid gracilis is actually more resistant to copper than most other metals. So a lot of these include things like cadmium, in my case, mercury, arsenic, lead. It has been surveyed across different species, especially with chlorella and clitomonas. Those are the two main model organisms outside of euglenoids that receive the most attention for metal work.
I agree. Copper is the exception. Everything else works fine with euglena.
Exactly.
Oh, somebody threw in, what about the other hundreds of euglenoids though? So what else are we exploring?
I mean, of course, like there's so many. Definitely Euglenoid gracilis is the model organism. I think everyone can kind of agree on that. But even with there's no exception in the metal biogeochemistry and remediation work. Mutabilis has also received a large bracket of that attention. But as far to the best of my knowledge, it's quite minimal outside of those two.
Right, and another problem with that is that many of those are cannot really grow outside their normal habitat. There's only one description, one in the literature, and you never heard about it again.
All right. If there are no more questions, unless there are any other questions, and we want to throw them in the chat. Scott, do we want to take a brief break before? Oh, Alistair's got a question. We can jump on in.
Yeah. Hi, it's Alistair Simpson here. I was the guy asking those annoying questions in the chat. Sorry, this isn't my field at all, but wouldn't it be like awfully coincidental if the species that had the highest tolerance for heavy metals within euglenoids just happened to be the one that's a attractable model organism for completely unrelated reasons?
Actually, I can speak to that really quickly. I'll jump in because this is exactly what my research is on. I'm not looking at gracilis, I'm looking at Mutabilis, and I am looking at it from a mixed culture that was isolated from a mine in Northern Ontario. So it was naturally found in a pH of about 1.5 and absolutely riddled with toxic concentrations of heavy metals. And what I've noticed in my research when I've compared it to a colleague that actually looked at Euglenoid gracilis is exactly what Vaughn said. When you're challenging it with cadmium, Mutabilis did have higher resistance, especially in that fungal and bacterial co-culture. So the inability to be able to isolate the organism probably has something to do with that tolerance. And if you actually do a quick Google search of Mutabilis versus gracilis, you're going to find that Mutabilis is incredibly under-researched. Vaughn has some of the more recent papers that are coming out about it, but besides that, there's some groups in Europe that were looking at trying to sequence the chloroplast genome back in 2016. It's just not a super heavily studied organism. So part of what I'm trying to do is tilt the tables a little bit and say, well, everybody's studying gracilis, but maybe I'll make everybody turn their heads a little and look at the organism that actually is more tolerant to everything except copper now, as we know. So it's just it's a happy coincidence. Yeah.
May I ask another question for that? How difficult is it to grow Mutabilis?
How do I grow it? I grow it the same.
No. It is complicated.
It's slow.
Very slow. It's very slow. My experiment was on average double the length of my colleagues that were working on gracilis. So if you do we tried to suppress fungal and bacterial growth with antibiotics and antibiotics, it grows even slower if it grows at all. Many of them just kill it. Right. So
this might be the major difference. I mean,
yeah. It's definitely a slow.
Gracilis is kind of weed. It grows everywhere and very fast.
And one of the interesting points with the metal resistance is that we don't know why. I mean, to this point, like, I've kind of alluded to maybe it's the metabolite pool that's being released, but nobody's really looked at this fungal interaction with gracilis. Maybe it's the fungi, maybe it's not actually gracilis at all, but rather this like symbiotic relationship or scaffold between these euglena that provides a physical barrier to help minimize the amount of surface interactions. It's interesting. I'm not sure, but
Yeah, and so Vaughn, what I'll be doing is pretty much what your paper was doing, where I'm looking at transcriptome analysis and metabolomics in the whole system and seeing if I can relate it to gene expression in the different organisms.
Oh, wonderful.
Maybe one more fun fact. It's that grassroots is very resistant to radiation. So we are talking about 9,000 gray, and they are not impaired at all. This is crazy.
There's some fun organisms.
Yeah, right.
Can you isolate the bacteria or fungi? Like,
yes, of
I
course.
haven't been able to, no. I haven't been able to isolate anything.
okay.
But Emma, you were able to select conditions in which you could get increased growth of one of the components versus the other. Is that not correct?
So very similar to what Celine was saying earlier about adding the carbon source, growing it in a carbon source media, you see the takeover
Which
of
one?
the fungus.
Which one are you using?
Triptych toy broth.
Okay.
It was just sitting around the lab, and I talked Barry into letting me
Any
just do
further
some fun
suggestions on that,
experiments.
Michael?
Yeah, no, we are using the acetate. So this is quickly eaten up, and so it's gone. But it shouldn't be any problem. I mean, you ever consider to do some metagenomics?
That's the plan.
Oh, okay.
Yeah,
Okay,
we just have
so
to doing some pack bio sequencing, long read sequencing. It's just getting it to isolate the DNA nicely, because it's such a messy culture that getting some good purity ratings is a little difficult. So I'm in the process of working on that and sending the samples off to Dalhousie University in the east coast of Canada.
Because we are trying to apply that for microbiomes, in internal microbiomes and external microbiomes. It's the only way. Everything else doesn't work.
Yeah, we don't really even still know what's in there, and that's where my fellow colleagues in the Emory lab all kind of shook me last year and said, Emma, how could you even be working on something when you don't actually know what's in there? So we had to pivot a little bit and figure that out.
Unfair. First. This is very unfair.
I know.
There are some, I'll bring up a collaboration opportunity, because there are some Alliance International grants that sounds like there's some mutual interest in this topic. Canada has some grants that you might be able to apply for to do something together. So just throwing that out there.
Thanks,
sounds like fun, yeah?
I'm here for 3 more years, hopefully. So if we don't have any more questions, if we wanted to chat in the meantime, I believe in 5 minutes, Scott, we've got the annual general meeting. Otherwise, everybody else is off the hook. So great session today. I appreciate the stimulating conversation for sure.
Awesome. Yeah. So thanks a lot to all the presenters. Session chair Barry, that was a wonderful chairing session. Thanks a lot. And then Emma, fantastic as always. And thanks to all the presenters for their great contributions. So I'd ask the EIN Executive Advisory Science Committee and people that helped put together the position paper to hang around. We're going to be having the annual general meeting now, but everybody else, please feel free to leave, and we'll see you tomorrow.